All right, we're starting a new chapter. It's on microbial metabolism. And our objectives for this part one of the notes are that you should be able to compare and contrast anabolic and catabolic reactions. Within this, we're going to explain the role of ATP as a coupling agent in these reactions and the role of enzymes in these chemical reactions. Now, but metabolism comes from the word metabolin, which means change. And so the two big ways we're going to be changing molecules in these reactions is we're either going to try to join together smaller molecules to make large ones, or we're going to take a large molecule and break it down into smaller ones. So the sum total of your chemical reactions in an organism is considered metabolism. And these chemical reactions will either release energy or require energy. So metabolism is really a balancing act between these two main processes. And those two classes of chemical reactions are what we call catabolism, those that release energy, and anabolism, those that require energy. So we'll start first with the catabolic reactions. Catabolic reactions are the breakdown of larger, complex organic molecules into simpler ones. So it's a breakdown reaction, and this will release energy in the form of ATP through the breaking of those bonds. These are also called degradation reactions because you're degrading or breaking down a molecule. They are also called hydrolysis or hydrolytic reactions. Lysis means to break down. So sometimes we call these hydrolysis reactions because not only are we breaking chemical bonds by breaking a large molecule into a smaller one, but we have to add water. Add water to the reaction. Okay? So sometimes these are called exergonic because they're producing more energy than is consumed. And a lot of times some of that extra energy is released in the form of heat energy. So they're also considered exothermic reactions because the therm deals with the heat that's being released at the same time. And so cellular respiration is a good example of a catabolic reaction because we're taking this glucose here and we're breaking it down with the help of oxygen, and we're releasing carbon dioxide, water, but what we're releasing is all of this energy in the form of ATP, okay? And that's cellular respiration. We'll get to that in big time later on in this chapter. But these molecules then, these simpler molecules, are the building blocks for anabolic reactions, okay? It also, catabolism provides the energy needed for the anabolic reactions. And so we really do couple these reactions together. Catabolic reactions help anabolic reactions. So anabolism is the building of complex organic molecules from smaller ones. So now we're joining together smaller molecules to form a larger, more complex organic molecule. But in order to do that, we require some input of energy in the form of ATP because we're forming those bonds. These are also called biosynthesis reactions or dehydration synthesis reactions. Remember, synthesis means to build. So we're building a larger molecule from smaller ones. So in order to do that, we have to release some water, okay? Because we have to remove the H and OH ends off of the molecules that you're going to join together. These are energonic because they consume more energy than they produce, also called endothermic reactions. So some examples could be if you were joining together some amino acids to form a protein, joining some nucleotides together to form like DNA, joining some simpler sugars to form a more complex polysaccharide. All of those are examples of that. But basically, this is how you generate the materials you need for cellular growth. We're always having to build, each time your cells divide, they have to make all those molecules, all those cell parts that are in there. Well, how does that building occur? Through these anabolic reactions. So, since we're talking about all either releasing ATP or using ATP, what is ATP? Again, it stands for adenosine triphosphate, okay? Because it's made up of adenosine, a ribose molecule, and 3 phosphate groups. That's where the triphosphate comes from. When that last or terminal phosphate is split, last just means terminal just means last, that's when the energy is released, and then you're just left with ADP or adenosine diphosphate. So anabolic reactions are coupled with ATP breakdown because you're using the energy at that point to join those smaller molecules to make a larger molecule. Catabolic reactions are coupled with ATP synthesis because if you're breaking a large molecule down into smaller ones, you're releasing that ATP. And then as a reminder, when we're breaking down large molecules into smaller molecules in catabolism, a lot of that energy may be lost as heat. So not all of it is used to make ATP. Some of it just gets lost as heat. But we need these reactions because we do need a constant supply of energy. If you did not have a constant supply of ATP for your cells, they would not function. And if they don't function, guess what? You won't function either. If your cells die, you die. So we really need that constant supply of energy. Again, this just shows you the role of ATP in both the anabolic and catabolic reactions. So if we start here with complex molecules, and we're going to break them down into simple molecules, that's a catabolic reaction. And so that's going to release ATP. It's going to give it off, okay? And a lot of it's going to get lost as heat as well. Now that we have these simple molecules, they can be joined together to form complex ones in an anabolic reaction. Well, that's going to take some energy here. It's going to take some ATP. So we're going to break off that last phosphate group and use that energy from the ATP, and then we're just left with ADP here. Catabolic and anabolic reactions are all, they can only occur if there are enzymes involved, okay? And each specific metabolic reaction has a different enzyme that's causing it to occur. And again, these enzymes are encoded by your genes because enzymes are proteins. And so your DNA is what determines your proteins that are made, thus your enzymes as well, which play a role in your metabolic reactions. And just a little bit of collision theory is just stating that chemical reactions occur when atoms, ions, or molecules collide. Well, it's the energy that's being transferred in these collisions that either break chemical bonds or form new bonds. And so activation energy, important one you're going to need to know, that's your energy that you basically need to start a chemical reaction. It's enough energy to get that, disrupt those electronic configurations, and so that you can get either those bonds being broken or those bonds being formed. But it's the energy needed to start a chemical reaction. And then the reaction rate is just the frequency by which those collisions occur so that they can bring about that reaction. And so what we can do to increase that reaction rate is include enzymes or increasing temperature or pressure. Well, can you increase temperature and pressure in your cells to make a reaction occur? No, that would destroy your cells. And so that's why we have enzymes, because enzymes allow our reactions to occur at cellular temperatures. So enzymes are what we call biological catalysts. That means they speed up chemical reactions without being used up in the process. They are specific. They only act on a specific substance or substrate. So every single reaction in your body has a separate enzyme that's playing a role in it. Enzymes have what are called an active site, and that's the part that interacts with the substrate. That forms the enzyme-substrate complex so that the chemical reaction can take place. Enzymes lower the amount of activation energy needed to start a chemical reaction. Why is that important? Again, we can't just use heat. If we were to go into the lab room, we could fire up a Bunsen burner or a hot plate and we could heat up our substance and get that reaction going. You can't do that to your cells. You can't just heat them up. Otherwise, you would destroy the proteins. They would denature and it just would destroy your cell. So we use enzymes because they lower the amount of activation needed to start the reaction, which means then all of your reactions can occur at cellular temperatures. Okay? That's what we want. We want all of our reactions to occur at our own body temperature. And so the parts that I've starred here are probably the most important things for you to remember about enzymes. They do usually end in ASE, not all of them, but most of them do. So that will give you a clue if you're looking at an example, if it ends in that's an example of an enzyme. And this is just showing you the reaction. If we're starting out with your reactants here at the beginning and we're trying to produce these products here at the end, here's what a normal reaction, how much activation energy you'd have to put in to get to that end product. That's a lot of activation energy. Now, if you start here and you have an enzyme involved, look at how much less activation energy you had to put in. Almost half the amount. Okay? That's what enzymes do for us, is they lower the hill, basically. They lower the activation energy needed to start that chemical reaction going. And here are just some samplings of some different enzymes. Notice they all end in ASE, but they all have different jobs because every single chemical reaction in your body has a different enzyme. So I won't require you to memorize all these, but should you go on to AP Bio or some of your college courses, you would have to know all of these. You might remember ligase when we talk about DNA replication. After the polymerase goes down and puts down the new nucleotides, ligase comes along and seals up everything. Here it shows you an enzyme right here, and here's the active site. Okay, that's the spot that the substrate here is going to fit in, and only since it has a very specific shape, only this specific substrate can attach to it. That's why enzymes are so specific. Once that substrate attaches to it, we call it the enzyme-substrate complex, and now the chemical reaction can proceed. In this case, it looks like we're taking one large molecule, and it looks like we're going to break it down into two smaller products. But notice here, the enzyme did not change at all from this beginning picture. It does not get used up or changed in the process, remember? It stays the same, so that's nice. We can use those enzymes over and over again. Some enzymes are just entirely proteins, but most enzymes have what's called an apoenzyme, which is the protein part, and then a cofactor of some sort, which is the non-protein component. Some of this is a coenzyme, which means it's an organic cofactor. And so when we add those together, we call it a holoenzyme. It's the apoenzyme plus the cofactor, which makes up the whole active enzyme, or whole enzyme. So here you have, here's the apoenzyme, but then once you add the coenzyme to it here, it kind of rounds out the rest of the active site here for that substrate to attach to. Some of the coenzymes are some of the electron carriers that we're going to be talking about in cellular respiration. So a lot of these here, NAD plus, NADPH, FAD, once they grab onto the protons, they're going to become NADH, NADPH, FADH2. They'll all be carrying those electrons to the electron transport chain. Coenzyme A is going to play a role in the Krebs cycle, so we'll talk about that a little bit later. But many of those coenzymes are derived from vitamins. And that's the end of this part of the notes.